Thanks, everyone, for attending. This is a very exciting moment for me to be able to introduce my dear friend and former colleague, Jean Fan, who is coming to us virtually from Johns Hopkins. Actually, which, yeah, you're associated with a bunch of different places. So is Center for Computational Biology part of the Biomedical Engineering department?
Yes.
Okay, all right. So officially you're in the bioengineering department at the Center for Computational Biology. Jean started her career at Johns Hopkins, so I guess you kind of came full circle. So as an undergrad, she studied applied math and biomedical engineering, and then went on to do a Ph.D. at Harvard in bioinformatics and integrative genomics at Harvard Medical School, where she worked with Peter Karchenko on developing a bunch of really cool new ways to look at single-cell RNA sequencing data. And after a very successful and extremely productive PhD, Jean joined Xiaowei Zhuang's lab at Harvard University Department of Chemistry, where I was doing my postdoc at the time. And Jean was sort of a force of nature who just like swept through the lab and changed the way we did image analysis and all sorts of analysis, and really spearheaded the way we looked at MERFISH data. And I was very fortunate to be Jean's neighbor. And so I've learned a lot from just few, I guess, how long was it? Like a year and a half that we shared an office?
Yeah, I guess before COVID hit and drove us apart.
Right. It's okay. I hope that you'll be here on site and visit soon. But yeah, there was a wonderful time to be able to learn from Jean. Jean is an incredibly talented and extremely organized and deliberate scientist in a way that I aspire to be one day. And she has been recognized, I mean, first and foremost, from all the beautiful papers she's written. She's also won the Nature Research Award for Inspiring Science. She's been named as a Forbes 30 under 30, which may or may not still be the case, but I'm not going to talk about age. Actually, you're probably still under 30. Jean is very successful very early in life. She has also been very successful outside of science. For those of you who are not aware of this, Jean is an accomplished photographer, and she's actually produced many book covers, among other things. She's also given a TED Talk that has the title, "Do Art Like a Science, Do Science Like an Art." And I think that title already says a lot, and I would recommend everyone to take a look. And the thing that I actually find the most inspiring about what you've done is she's led this initiative called Customized, which is a publishing initiative where she's created children's books that can be personalized. So you can basically order these books to look like your child, and to where the main character in these books have the name of your child. And then these children, or your children or children of someone else, can read about sort of fictional versions of themselves where they are successful in different STEM fields. And I just think this is a phenomenal idea. These books have been over 20,000 copies. I don't know if that's the latest number. How many books have you published as of yet?
I think the latest stats may have been double that. Yeah, probably over 40,000 now.
So at least 40,000 children have been inspired by Jean's books. So it's customized with STEM in the middle, if you're curious about it. Really cool organization. Jean, you're an inspiration to not just children, but to me as well, and hopefully a lot of other scientists. I can't wait to hear what you've been up to for the last year as a faculty member, and I'll give the floor to you. Oh, and if anyone has questions, please type them in the chat box. And if there's something you need clarified, I'll just interrupt Jean and I'll read your question. And if not, we'll wait to the end, but just type it in the chat, not in the Q&A, the chat. All right. Thanks.
Yeah, well, thanks so much for the really wonderful introduction, Paul. I'm so flattered. And yeah, definitely hope to, one day be able to meet up in person again. But for now, I'm very excited to be here with all of you virtually, and special thanks also to Peter for the invitation, and bring us together virtually. So today I'm very excited to be sharing with you some of the latest work coming out of my group. I started my lab at Johns Hopkins during the midst of the pandemic, I suppose, in July of 2020. So it's been a little over a year now. And actually only recently did I meet my students in person. So it's a bit of an interesting way to start a new lab, I suppose. But yeah, today I'm going to try to share with you all a story about learning about from other disciplines and also from each other in the way we've developed computational approaches for analyzing spatially resolved transcriptomics data. Okay, great. So then, yeah, my lab is very broadly interested in understanding how spatial contextual mechanisms impact cellular heterogeneity at both the subcellular and the tissue level. So particularly at the tissue level, for example, we want to understand how are cell types and cell states organized within tissues. Particularly, we're interested in certain diseases such as cancer, particularly these pediatric gliomas and glioblastomas. But generally in cancers, we have genetically and transcriptionally distinct neoplastic subpopulations that may actually differentially interact with different immune cells or other cell types in the tumor microenvironment to promote therapeutic resistance, angiogenesis, and ultimately impact the clinical outcomes. So we want to understand or better understand how this spatial organization of these different cell types is related to tissue function in both the healthy setting and also in this the progression of various diseases like cancer. And I think as multiple speakers in this series have already really beautifully highlighted and demonstrated that to address these biological questions, we would really benefit from spatially resolved transcriptomic technologies. I think you've already heard from Jeff Moffitt and others to explain the kind of chemistry behind MERFISH, but there are a number of other also diverse spatially resolved transcriptomic technologies that perhaps differ a little bit in their chemistries and their capacities, but effectively they all allow us to profile in a very high throughput manner the transcriptional states of these individual cells or even small groups of cells in a very high throughput manner while maintaining the original spatial organization of these cells or small groups of cells within fixed tissues. So then, with this new technology and the measurements that it enables, we can begin asking our biological questions of interest. But of course, to do that, we need not only the technologies to generate this data, but also the new computational methods capable of taking advantage of this new spatial information. So my lab currently focuses on developing these computational methods. My student, Laila Ada, and I here recently discussed some of the challenges and opportunities associated with these computational methods in our recent Nature Communications commentary. So please do check that out if you're interested in learning more. But broadly speaking, we find that current spatially resolved transcriptomic technologies tend to fall in these two camps. Those based on high throughput in situ targeted transcriptomic imaging of pre-selected RNAs that is able to give these types of spatially resolved transcriptomic measurements at molecular or single cell resolution. So MERFISH would be one of these. But also the other camp that we see is those technologies based on spatially resolved RNA capture, followed by sequencing for typically non-targeted kind of genome-wide transcriptional profiling, but at pixel resolution. So the suitability of each of these camps of transcriptomic or spatial transcriptomic technologies, or even the specific technology itself, will very likely depend on your particular biological question of interest and specifically your particular needs to kind of balance the experimental throughput versus the spatial resolution. But of course, then, given these two camps of technologies, they also present us with different computational challenges in terms of the downstream analysis. So for the rest of this talk, I'll essentially try to teach you guys about some of these analytical approaches that my lab has been developing to address both of these types, the challenges in both of these types of data. Okay, so first, focusing on targeted transcriptomic imaging technologies. As you might already know, what these technologies, including MERFISH, allows us to do is image effectively, what RNAs are within the cell. So, for example, after many rounds of imaging, we are able to essentially decode each point here is an RNA, and we can tell which ones correspond to a particular gene species. And of course, we can do that not only for one or two genes, but now hundreds to actually up to 10,000 different gene species. And of course, we want to do this not only for one cell, but we can do it for hundreds of thousands to now millions of cells within fixed tissues. And then what we can do is segment these cells and count, how many of each RNA species is in each cell, and that gives us effectively, single cell resolution, spatially resolved transcriptomic profiling for these targeted gene species of interest. So then, what we get at the end is not only an image here of the cortical section of the mouse cortex, coronal section of the mouse cortex, where we can see each point is a single cell, but we can actually also now know how much of each various genes are expressed within the cells within the tissue. So here, we have excitatory neuronal marker where red is high expression, blue is low expression, and you can just visually begin appreciating the spatial organization of these gene expression patterns. Likewise, GAD1 is an inhibitory marker, and so on and so forth. So we can see all these beautiful different expression patterns. And further, we can perform dimensionality reduction and clustering analysis as we do with non-spatially resolved single cell transcriptomics data. Hopefully my animation comes through. It might be a little choppy, so I'll let it play a few times. But what I'm showing here is clustering of these single cells based on their transcriptional measurements to identify transcriptionally distinct cell types. But of course, now we have the original spatial positions of these cells also, so we can further explore how they're spatially organized within the original tissue. And understandably, for the brain, we can see how these cell types are very spatially organized. So expectedly, if we were to look for genes that were spatially organized, we would expect the genes that mark these different cell types to also be spatially organized. However, if we were to focus on an individual cell type, such as here, the pyramidal layers of the hippocampus, one question that we had is, can we identify additional gene expression heterogeneity within cell types that were coordinated in some spatial manner? So this is the question we sought to tackle with Brenda Miller in my lab, with a lot of insightful input from DJ and Catherine Duloc in the biology. Is how can we characterize spatial gene expression heterogeneity within cell types and tissues? Particularly, we were interested in doing this in a manner that was robust to non-uniform cell densities, because as you saw, the pyramidal cells in the hippocampus, it's not a uniform distribution of cell densities by any means. So we want to make sure that we're not accidentally mistaking spatial variation in cell density, for example, for a spatial variation in gene expression. So we developed this approach called MERINGUE. And for more details on the method, it actually just came out in the October issue of Genome Research. So please do check out our paper for more details, as well as our lab website for our software package and additional tutorials if you're interested in trying it out. So what MERINGUE does is actually we draw inspiration from geospatial analysis, and specifically we build on an spatial autocorrelation analysis commonly used in geospatial data analysis, where there is a statistic called Moran's I that's previously been used to evaluate whether particular quantities, say here is shown income, exhibit non-random spatial variation. So in this case on the left here, even though this kind of looks like a tissue maybe, but actually each spot here is a county, and this is a particular geospatial map where we've colored the different counties or neighborhoods by their income level. And as you can see, just visually, income is not randomly distributed. Income of neighboring counties tend to be correlated. So if this is a county that has high income, it tends to be spatially adjacent to another county that's high income. So then we can quantify income and its level of spatial autocorrelation using Moran's I and evaluate the statistical significance of that. And we can do this for other quantities as well, in addition to income. But in our case, of course, we're not interested in necessarily income in counties, but we are interested in gene expression in cells. So we can do something very similar, where we can essentially look and ask if a cell's expression of a particular gene is highly correlated with that of its spatial neighbors. That gene would essentially have high spatial autocorrelation, and we can then quantify that using Moran's I, and in that way use Moran's I to distinguish genes that are highly variable in a spatially coordinated manner, such as the two cartoon genes shown on top, versus genes that are variable but perhaps not in a spatially coordinated manner, like the gene shown on the bottom. So then what does it mean for cells to be neighbors? Well, MERINGUE uses a Voronoi tessellation-based approach for identifying spatial neighbors. It's also sometimes called Delaney triangulation. But effectively, in this way of encoding spatial relationships, we can represent each cell as a neighborhood, and the neighborhoods can vary in size. But two cells are considered spatial neighbors if their neighborhoods share an edge. This particular way of defining spatial neighbors and neighbor relationships between cells is very flexible. So we can actually do it in 2D or 3D, or take advantage of multi-section data. But for us, we were interested in using it because it was also robust to deformations in cellular density. So what this means is that if we take something here as the olfactory bulb, where each point is a group of cells, and red is high expression, blue is low expression for a particular gene. But what you can see is, we can have a uniform density case where all the cells are uniformly have uniform density across the tissue. Or we can simulate an example where we now warp the cell position so that we induce non-uniformity. And if we use MERINGUE and this Voronoi tessellation-based approach to encode the spatial relationships, the two ways of, under both the uniform and non-uniform density case, the neighbor relationships are pretty stable. And as a result, if you were to look at, let's say, the negative log 10 adjusted p-values of all the genes detected as spatially variable under both the uniform case and the non-uniform case, you see a very nice linear relationship, or a high correlation, which basically suggests that, regardless of whether we're in the uniform density or the non-uniform density case, we can still identify the same set of spatially heterogeneous genes. In contrast, this is not the case if we were to, there are other approaches that are relying on Gaussian distances between cells. And expectedly, if you were to change the distances between cells, you also impact the ability of these methods to detect spatially variable genes. Okay, but not all spatially heterogeneous genes or gene expression patterns are equal. In biology, we're often interested in spatial patterns that are not just driven by hotspots, which could arise, frankly, from minor errors in clustering, or alternatively technical artifacts. So MERINGUE builds on this other geospatial statistic called a local indicator of spatial autocorrelation, or LISA, to characterize the scale of spatial patterns. So we can show, for example, that if we were to just randomly shuffle gene expression, we expectedly, under the null distribution of a uniform null distribution, we still do get a 5% type 1 error rate. But if you were to look at the scale of these false positives in terms of their lisas and the size of these spatial patterns, they're actually all driven by a very small percentage of cells. So on the right is the distribution of the lisa scores for our false positive detection, spatially variable genes that we detected are false positives, and you can see that they're mostly quite small. So therefore, by requiring both significant spatial autocorrelation as well as for the spatial autocorrelation pattern to be driven by, let's say, more than 5% of cells, we can effectively eliminate false discoveries, which is quite useful when we're trying to prioritize genes, for example, for downstream validation. We just don't want to, we want to try to limit our false positives as much as possible.
So Jean, do you mind if I ask a quick question?
Yeah, of course.
Yeah, so in the Voronoi approach that you're using, so when you tessellate it, does distance, like actual spatial distance, not play a role at all? Like, can you have two networks or two cell patterns that have, if you just scale it, is it the same? Or if you take two cells and you like move them? So basically, what I'm asking from a biological point of view, like, are we discounting the role played by spatial distance in favor of like direct contact?
Yeah, it's a great question. In our case, we do have a filter, for example, to limit distances that are too large. So, for example, if we know there are certain paracrine signaling that we're interested in that can only take place within a certain micron distance, we can still restrict neighbor relationships to be those within a certain micron distance, for example. But that being said, I think you are right that if for example I were to, let's say, do expansion on a tissue and image it, if I were to not restrict the micron distances at all, the neighborhood or the neighbor relationships would be the same, because they in theory are not necessarily taking into consideration they don't necessarily have to take into consideration the distances themselves. It's the relative spatial positioning. Yeah, great questions. Let's see, well, so where was I? Okay, great. So in our paper, we apply meringue to many organisms, technologies, and in particular, the Moran's-I equation and the way we define spatial neighbors is quite flexible. So it allows us to, for example, identify spatially heterogeneous genes for cells that are truly in 3D. But we can also look at multi-section data where we have consecutive sections of the same tissue and specifically look for spatial autocorrelation that spans across tissue sections. So that are spatially autocorrelated, in Z. And we can actually also even look at specific receptor ligand pairs among spatially co-localized cell types to potentially look for cell-cell communication. And that's, I think, for the sake of time, that's the one story I'll try to tell you guys a bit more about. And this might be a data set you've already seen from previous speakers, where this is the mouse preoptic region assayed by Jeff Moffitt and DJ Malbamaku, where they applied MERFISH to characterize various cell types within this particular region of the brain, which has been shown to control various social behaviors like parenting, mating, aggression, and so forth. So Jeff and DJ were able to previously do a lot of beautiful analyses, identifying different cell types and probing the spatial organization of those cell types from the anterior to the posterior mPOA as seen here. So we essentially wanted to ask a follow-up where, even within cell types, can we identify additional spatial gene expression heterogeneity? And particularly, we wanted to look for patterns that were consistent across tissue sections and reproducible across animals. So this ends up being, a large number of statistical tests that, it's good to have a framework for doing, because we ended up analyzing, 70 different cell types across 12 different tissue sections, each, for over a dozen animals. And indeed, we were able to identify some aspects of spatial gene expression heterogeneity within cell types in the mPOA that were consistent across sections and also reproducible across animals. So for example, here, what I'm showing, each point is a cell, and the cells of inhibitory subtype I8 are highlighted. Since these are inhibitory cells, understandably, they express GAD1 pretty highly in red. So GAD1 isn't spatially heterogeneous because it's pretty uniformly expressed. But then there are other genes, including calcium binding proteins and secreted glycoproteins that may play roles in neuronal guidance. We also have neurotransmitter receptors and so forth that do seem to have a spatially coordinated variation in their gene expression. So for example, being more highly expressed towards the higher regions and more lowly expressed towards the lower regions, or more highly expressed towards the midlines. And this is a spatial pattern that's also reproducible across animals and also consistent across tissue sections. So interestingly, not all the spatial patterns were consistent across animals. And particularly, we did identify TACR1, which encodes for a particular neuropeptide receptor that's in inhibitory or excitatory subtype E8. We were able to quantify its spatial gene expression pattern. And what we found was that indeed it was consistent across tissues and everything, but it seemed to exhibit spatial aggregation only in females. So for example, as shown, or only in males, excuse me. So as you can see on top, even though this gene might be comparably variable in terms of the spatial variation, it's largely only in the males on the bottom, where it's kind of all red together or all blue together, whereas in the females it's much more scattered. And of course, we can look at it visually, but we can also quantify it using our Lisa statistic and be able to show that on a statistical level that we can see a sexually dimorphic gene expression spatial variation for this particular gene and this particular subtype. So yeah, given that the mPOA has been shown to control for essential social behaviors such as parenting, mating, and aggression, and other sexually dimorphic behaviors, it's also quite interesting to see that perhaps these sexually dimorphic behaviors could be encoded not only in the cell type differences, but within or the cell type abundances or their activations, but also the spatial organization of these neuronal subtypes that could be indicative of interactions with other regions of the brain, for example. Okay, so finally, building on Moran's eye, as I mentioned, the equation is quite flexible. So in addition to looking at, for example, all neighbors, we can actually limit, for example, to only neighbors, spatial neighbors that are of different cell types. So for example, here we can consider cells only neighbors if they're next to each other and one is cell type A and one cell is cell type B. And in this manner, perhaps we can look specifically for pairs of genes, for example, such as gene A, which is highly expressed in cell type A, and gene B, which is highly expressed in cell type B. Perhaps this is a receptor, perhaps this is a ligand. But if we were to just look at the gene expression correlations of these two genes in all cells, of course, they would be negatively correlated. But perhaps even visually, you can tell that the cell type A that express gene A tends to be higher. The cells of cell type A that express gene A highly tend to be spatially neighboring cells of cell type B that express gene B highly. So we can specifically look for these spatial cross correlations between spatially adjacent cell types, and in this manner, perhaps identify receptor ligand pairs that could be indicative of cell-cell communication via this particular receptor ligand interaction. So we essentially adapted the Moran's eye statistic to look for this, and we applied it to the mPOA data. And interestingly, we were able to find the significant spatial cross-correlation between aromatase, which is encoded by this CYP19A1 gene, and specifically its expression in neuronal subtype I2 is spatially cross-correlated with estrogen receptor expression in the spatially neighboring cells. So that was the one that was reproducible across animals and consistent across tissue sections also. Interestingly, we don't see the same type of spatial organization for example, for aromatase and androgen receptors in I2 cells in the same way. Just as a little background, aromatase is an enzyme that converts testosterone to estrogen, thereby modulating the steroid hormone signaling in the preoptic region. So testosterone typically binds androgen receptors, and estrogen binds estrogen receptors. So perhaps the spatial organization of this aromatase expressing neuronal subtype I2 with the ESR, with the estrogen receptor expressing neurons and their spatial neighbors suggests that the estrogen synthesized by these I2 aromatase expressing neurons may be interacting with estrogen receptors and the spatially adjacent ESR1 expressing neurons in a paracrine manner. So this is just kind of, I think, another cool way to demonstrate how we can integrate and use spatial organization to identify interesting spatial patterns that could be relevant to how the spatial organization ultimately relates to functional consequences. So yeah, just getting back to now our hippocampal, pyramidal cells, we can of course apply MERINGUE. And in this case, we indeed identify other multiple genes that exhibit spatial gene expression heterogeneity that are coordinated in a spatial manner, a few of which are just shown here. And yeah, if you're familiar with the hippocampus, perhaps you can just already visually tell that these spatial patterns somewhat, at least visually, seem to correspond with the different regions of the hippocampus, different hippocampal subfields specifically, like CI1, CI2, CI3. Yeah, so we're currently actually trying to develop more quantitative approaches to actually map these molecular transcriptomic profiles into larger atlases, such as the Allen Brain Atlas in 3D, so that we can better understand the molecular underpinnings of these different brain regions, but also then integrate what we know about these different brain regions and their inputs and projections, for example, to then enhance our understanding of these observed molecular transcriptional differences that we see in common cell types. Yeah, so this is work with Manju Anand and my group, with Daniel Torrey and Mike Miller at Hopkins. And yeah, we have a conference preceding paper that you're welcome to check out for some more info on that. And yeah, so then for more information on MERINGUE, definitely you'll feel free to check out our manuscript and also our lab website, which has lots of additional tutorials in case you're interested in trying this analysis out on your own. Okay, so now to switch gears a little bit, the second camp of spatially resolved transcriptomics technologies I mentioned is based on these spatially resolved RNA capture followed by sequencing. And these methods face some additional unique computational challenges, mainly related to the fact that depending on the tissue you're studying, the organism being studied and the size of each spatially resolved pixel or spot, each of these pixels can contain multiple cells. So this is just shown here. If you look on the right, if we zoom into this particular set of spots, you can see from the underlying H&E image that there are multiple cells within each spot, which means the spatially resolved transcriptomic measurements that we get is the aggregate of all the cells within the spot, which in theory could represent multiple cell types, which would then confound our ability to discover cell type specific spatial organizational patterns and also cell type specific transcriptional changes. So Brendan Miller in my group, along with a number of other folks, in particular with Yang Huang, Laila Ada, and Alton Sahu, they sought out to ask, can we recover cell type specific spatial organizational patterns and gene expression profiles from these multicellular, pixel resolution, spatially resolved transcriptomics data? And particularly, we were interested in developing an approach that didn't rely on single cell references. So we developed an approach called ST-deconvolved. And in particular, we wanted to be not limited to samples where we could get matched single cell references, because we were interested in just various situations where either due to biological, technical, or other potentially unknown perturbation-specific variations, we weren't able to get that perfect reference. And I'll try to elaborate on that in a bit. So as I mentioned, we developed an approach called the ST-deconvolved. It actually draws inspiration from natural language processing, where a generative modeling approach called Latent Dirichlet Allocation has been commonly used for natural language topic modeling. And in topic modeling, for example, we often have this collection of documents, say news articles, and these news articles are composed of words, say ball and microscope or something. And these words represent latent topics like sports and science. So we can model each document as a distribution or a mixture of topics that are distributed over these words. And we can then deconvolve the proportion of each topic within each document, as well as the association of each word with each topic. So then, hopefully you can see some parallels there that, we're not interested in topics per se, but we can think of each multicellular spatially resolved measurement as a collection of composed of different genes that represent mixtures of different cell types that are distributed over distributed over different genes. So then we can effectively deconvolve the proportion of each cell type within each pixel, as well as the association of each gene with each cell type. So there's some additional, feature selection and other estimations of optimal number of cell types that ST-deconvolved does that I won't really get into, but please do check out our bioRxiv preprint if you're interested in learning more. So just as a first proof of concept, we actually took the MERFISH data of the mPOA and we simulated pixel resolution spatially resolved transcriptomics data by essentially taking 100 micron spots and then aggregating all the cells and their transcriptional profiles. And then we asked, can ST-deconvolved recover the cell type proportions and cell type specific gene expression profiles? And indeed, we can. And just visually, it matches up pretty well, but we can also quantify it based on root mean square error of the cell type proportions, and also look at the correlation between the deconvolved gene expression profiles for the cell types and the ground truth gene expression. So we can see that we can do pretty well, and we can actually also compare our method to existing supervised approaches and show that we're able to offer pretty competitive performance. So I do want to quickly emphasize here that deconvolution is distinct from clustering analysis, as I showed previously. We can take cells and we can cluster them into transcriptionally distinct cell types. But here, we can have instances where deconvolution and clustering give different results. So as a proof of concept or a simulation, we took 3 different cell types from single cell sequencing and mixed them in different proportions. So that we have, some pixels that are mixtures of luminal cells and dendritic cells, and others that are mixtures of dendritic cells and parasites. Something that we can often see in spatial transcriptomics. If we were to just perform clustering analysis, understandably we get 2 clusters, neither of which represent a distinct cell type per se, but rather, the 2 various mixtures of the 2 cell types. However, if we were to do deconvolution with ST-deconvolved, we actually can recover the original spatial organization of the 3 cell types, as well as their individual transcriptional profiles. So that is how, one way in which deconvolution is distinct from clustering analysis. And we can actually see this in real data as well, where we look at clustering analysis of multicellular spatially resolved transcriptomics data of the mouse olfactory bulb here. Each point is a 100 micron pixel, which has many cells, and we can take the measurements of the gene expression of all the cells within pixels and do various clustering analysis and identify 5 transcriptionally distinct clusters that generally represent, that match our understanding of how layers are organized within the mouse olfactory bulb. But we also know that based on prior studies, there are many additional layers in the olfactory bulb. And in particular, we were interested in this, rostral migratory stream that would bring in these migrating neuroprogenitor cells towards, that should be towards the middle of the granule layer. So we applied SD-deconvolve, and we were able to pick up on a number of additional layers, including this deconvolved cell type 7 highlighted here, which just spatially seem to correspond to where we would expect the rostral migratory stream to be. And interestingly, if we were to then further look at what genes are associated with this deconvolved cell type, we indeed find genes like SOX11 and NRAP and other neuronal progenitor markers that if we then were to look at in situ hybridization stains of these genes, they indeed mark a much finer region anterior to the granule cell layer. So in this manner, we find that SD-deconvolved and other deconvolution approaches are able to recover cell type proportions and cell type specific gene expression profiles for these neurons within the rostral migratory stream that were not apparent from the clustering analysis alone. So then, perhaps circling back to one of our original motivations for doing this type of analysis in a reference-free way is because we really wanted to apply it in a cancer setting. And particularly in cancers, due to prevalent intratumoral and also intertumoral heterogeneity, it's hard for us to get multiple samples that perhaps have the same populations represented, especially given how precious and limited these samples are. So in that sense, we wanted to then apply SD-deconvolve to cancer data sets, and in this case, we applied it to breast cancer biopsies, where previously people were able to do, again, clustering analysis and show that they were able to identify 3 broad clusters of transcriptionally distinct spots that appeared to correspond with pathology annotations that are the ductal cells versus the non-invasive regions and the invasive neoplastic regions. But then applying SD-deconvolve, we can actually get many deconvolved cell types. One in particular that we were quite intrigued by was this deconvolved cell type 15 shown here. You can see it's a sub-proportion or a percentage of many different spots. But we were quite curious because it was spatially co-localized with or enriched within the invasive and cancerous regions. And if we were to look at what are the genes highly associated with this deconvolved cell type, we see that there are many immune gene markers, and if we were to use gene set enrichment analysis to try to see what pathways might be overrepresented by these genes upregulated in this deconvolved cell type, we get a lot of different immune pathways, such as T-cell activation. So in this manner, deconvolution was able to recover spatial organization of these immune signatures that may correspond with the spatial organization of immune cells within the ductal carcinomas here. And finally, just again, as a proof of concept, we were very curious about being able to distinguish between excluded versus infiltrated spatial organization of immune cells, which is relevant for, has certain implications in clinical prognosis for various cancers. So we applied a simulation-based approach where we mixed in immune cells with luminal cells in a spatially organized manner, such that on the left you can see, there are little pixels where we have various, immune infiltrates, whereas on the right we have the red, a large, dense, perhaps neoplastic cell cluster, whereas all the immune cells are excluded towards the periphery. And in this particular case, given this, these two distinct spatial organizations, SD-deconvolved is able to then recover the original spatial organization, as well as distinguish between this infiltrated and excluded case for the immune cells. So then if you're interested in trying out SD-deconvolved, please feel free to visit our lab website at jeff.works/SD-deconvolved, where we have our package available, along with various tutorials to help you to run this method also on your own multi-cellular pixel resolution, especially resolve transcriptomics data. Great. And then with that, I'd just like to thank very much my lab, in particular, Brendan Miller, Laila Ada, Arpan Sahu, and Peyang Huang and Manju Anat, whose work was highlighted here, as well as our funding resources, as well as, all of you for your attention. And if you're interested in collaborating with us or have any questions about our software, please do feel free to reach out. My email is here. You're also welcome to check out our lab website at jeff.works. And once in a blue moon, you can probably find me on Twitter at jeffworks. With that, thanks everyone, and happy to take any questions.
Great.
Thanks, Jane.
Yeah.
Maybe I can start off by, I have a question when it comes to the spatial heterogeneities that you pick up in the first part of the talk, where you were saying you could actually identify genes that are spatially regulated or have some spatial pattern to them that are non-random based on, right? How do those subpopulations appear in the UMAP if you looked at the same data but from a single cell clustering map?
Yeah, it's a great question. And just to to paraphrase the question. So essentially, if you were to do a clustering analysis again, perhaps a subclustering analysis where you take only those, let's say, I6 cells and cluster them again, can you identify, let's see, spatial or transcriptionally distinct subpopulations that correspond to the spatial patterns that we identify? Yeah. So sometimes you can. Sometimes if the spatial patterns are coordinated enough with each other in such a way, you can see, for example, if you were to do a UMAP, it tends to be less distinct than cell types, where on a UMAP it'll be like a really distinct cluster here and a really distinct cluster there. What we tend to see is it tends to be more of a blob, and there's kind of a gradient where the cells towards the left of the UMAP will express highly these spatial gradient genes, and the cells to the right will express more lowly these genes.
Which, I mean, if you look at single cell sequencing papers, there are lots of examples where the authors will segment clusters even though they're just two parts of a blob.
Yeah. So that is the case if the spatial patterns are coordinated. So let's say they're all up in the same group of cells or all down in the same group of cells. That's not always the case. Sometimes you actually have, let's say, a spatial gradient from, let's say, middle to out, and then a different spatial gradient from up to bottom. And I guess if you were to take the intersect of all those, you'd probably end up with 4 clusters. But
that would be less informative if you're trying to derive some biological conclusion from it, right?
Yeah. So I think it's a interesting philosophical question of what defines a distinct cell type, and whether these cell types are just the intersection of all these different spatial gradients.
All right, so I have some questions in the chat. I'll start with a question from Gerald Chun. Who says, Thank you, Jean. Visually stunning and beautiful. Do you have approaches to look at splice variants?
Yeah, thanks so much, Gerald. Yeah, good to perhaps virtually interact with you again. Gerald and I worked together during my PhD. Yeah, so yeah, thanks for the question. And for looking at splice variants, for MERFISH, if we had designed probes specifically to distinguish between splice variants, so perhaps if there's a canonical splice variant, let's say, that has a unique exon, we could design, the exon is really large, I guess. Then I think we could design probes to target that exon, and then we probably also want to distinguish it from the other isoforms. But in theory, in a targeted based approach, I think we could look at canonical splice variants. That being said, for sequencing based approaches, such as the second camp of spatially resolved transcriptomics measurements I mentioned that are non-targeted but based on sequencing, my sense is those are currently 3' only targeted sequencing, which wouldn't allow us to really distinguish between splice variants. I think it'd be really cool, frankly, for somebody to do bar-coded, spatially resolved long-read sequencing. Just a hint if anyone's interested. It's not an area that I have the expertise to pursue, but yeah, I would love to look at splice variants in a spatially resolved manner, especially cryptic splice sites for perhaps novel splice isoforms in the cancer setting. I think that'd be really cool. But currently, we don't have any computational approaches per se to be able to do it.
I thought you were going to talk more about the RNA velocity and splice forms, but I guess that'll be up to everyone to look into in your publications.
Yeah, too many new works, I guess. If you gave me another hour, I could talk about that.
I guess we'll have to invite you again. All right, so Annabelle Coles has a question on the deconvolution approach. Can you use deconvolution to distinguish cells that are in different states as opposed to different types of cells, I suppose?
Yeah, it's a really interesting question. So there are a couple underlying assumptions with that we try to get into in the supplementary, very long supplementary notes of our papers regarding the why or in what regime these deconvolution methods work. One of the underlying assumptions is that the cell types or cell states have to be variable in their proportion across pixels. So for example, you need to have some pixels where you have, let's say, 10% of this one cell type, and in other pixels, it's 30% of the cell type, and other pixels, it's 90% of that cell type. So it's that variation that we're leveraging to be able to distinguish between, to deconvolve. So in that sense, in my experience, cell types and tissues tend to have more of that spatial variation. And it's an assumption that I'm not entirely as confident about more subtler cell state differences. The other thing is, so LDA works intuitively, perhaps. It's a bi-clustering type of approach where we're trying to identify groups of genes that vary together. So in order to distinguish cell states, they would need to have a good number of transcriptional differences, which is typically more prevalent for very distinct cell types. So if your cell states are indeed transcriptionally distinct enough that we let's say they have a couple hundred differentially expressed genes, and of course the proportions of those cell states or of those cell states within pixels are quite variable, then I think you could. But my guess is that in most tissue settings, the primary axis of variation is going to be driven by cell types. Like the cell types are just more different from each other than the cell states. So those will still kind of be deconvolved first, because they're just much more variable and distinct.
So that would mean that, I mean, even with these deconvolution approaches, and I know Fei Chen come out with one of these methods too. Yeah. So even with these approaches, there are some data that you can only get from the fish-based methods that cannot be recovered. Like for instance, if you have if there's variation in the expression profile of a single cell, and that only occurs, very sparsely or even once, right? That's something you can never get from something where you have a collective.
Yeah, absolutely. It's a great observation, and something we highlight in our bioarchive preprint is actually for very rare cell types. Let's say cell types that are present at 1 to 5% within each pixel. Generally, deconvolution really struggles with those, regardless of supervised or unsupervised.
You said 1 to 5% within each pixel?
Yeah.
So that's, I was going to say, like, so I studied the heart, and we have, nerve cells in the heart that are much.
Heart is big. Your cells are big.
Yeah, but on the other hand, there are cell types that are much rarer than anything that would go to that level, right? So then that would be a hopeless thing to look for with pixel-based approaches. I mean, let's say
based on our benchmarks for very rare cell types, it's I think just mostly very difficult to be confident. The one exception is if the cell type is just really transcriptionally distinct. You have, let's say, a perfect marker, I think that's like the only situation where it might work out.
Yeah, so that might work. And what do you think about when the pixels get smaller, which they inevitably will, right?
It's a great question.
Get to a point where eventually the pixel-based approaches will be able to recapitulate the same kind of data as we can now for the image-based approaches, but full transcriptome. And in that case, will the image-based approaches be useless?
Oh, interesting take. We definitely see pixels getting smaller. I mean, even when we first started out, I think pixels were around the 50 to 100 micron range, and now with newer technologies, I think we're seeing as small as like 2 microns, 2 to the 10 micron range. I think Visium is in the 10 micron range now. What we've seen is even at the 10 micron range, we're still, and depending on, of course, tissue and organism, we can still have multiple cells and multiple cell types. By the time you get to 2 micron resolution, you really are subcellular. But at that point, I think I do think at that point, you're not really, you wouldn't be doing deconvolution analysis anymore. There's no multiple cell types to deconvolve anymore. Instead, I think the type of approach you would end up doing is an aggregation-based approach, where you're trying to aggregate the subcellular pixels into recapitulating the whole cells. But I don't know, where I don't think imaging-based approaches will go away is because so I don't think imaging approaches will become useless. I still really like imaging-based approaches because of the subcellular resolution. So even if we have 2 micron pixels, if you just take, let's say, a whole cell, the 2 micron pixels are sometimes going to be, let's say, in the nucleus. Sometimes they're going to be overlapping with the ER region. You can probably start doing deconvolution on a subcellular level, I guess, to try to get at like nuclear versus cytoplasmic or ER or something. I think the dominant signal there would still be cell type. But anyways.
Does the pixel deconvolution work less well if the cells are more heterogeneous? Does it assume a certain level of homogeneity between the cell types which constitute the pixels?
It's a great question. No, it actually works way better if it's heterogeneous. The more distinct, transcriptionally distinct co-localized cell types are, the easier it is.
I think if I understand you correctly, Peter, you mean like, for instance, in cancer, where you might even have cells that transcend what we typically think.
Yeah, I'm thinking of, in kind of disease states where, kind of order starts to break down, whether that's in cancer or perhaps in aging.
If you have less well-defined
Exactly.
types.
Right.
That's what you mean. Let's see. That's an interesting question. I think as long as there's still genes that are correlated together, they're varying together, perhaps no longer across canonical pathways or in the same way as what we see in healthy cell types, deconvolution I think would still work, which is actually I think an advantage of a unsupervised based approach. So Pallav, you mentioned Fei Chen's approach, which is actually an approach we're a fan of, where if you're able to get a single cell reference from the same exact tissue that you're profiling by spatial transcriptomics, we find reference-based approaches like Fei Chen's SD or RCTD, it works really well. But whenever your reference isn't perfectly matched to the spatially resolved transcriptomic measurement, if, for example, we have this one simulation where we have analysis where we have aged tissues, aged tissues where we have aged macrophages, and then aged macrophages are transcriptionally distinct from young macrophages, and if you were to use a reference that's taken from a young tissue, and you try to deconvolve this aged tissue, you end up actually getting certain incorrect results. Whereas if you were to deconvolve, just the aged tissue itself, you can see how the aged macrophages are transcriptionally distinct from young macrophages. So it's I think there are lots of perturbations that we might not always be aware of ahead of time, such as, let's say age, how important is age matching, how important is various other types of matching criteria between our spatial omics measurement and the reference that we're using to try to deconvolve it.
So I was going to ask you about whether you've done much looking at aging. So when you look at aged tissues, do you tend to see more what you might call changes in cell phenotype with age, or changes in cell organization and spatial relationships with age?
Oh, that's a really interesting question. We haven't, I think we don't have enough access to various aged tissues to be able to really answer that. But I think that would be a really interesting question if we're able to generate spatially resolved omics measurements for lots of different aged tissues, perhaps even in a real time, a real time course. So we have many different ages. I think that'd be very, very cool.
Okay, well maybe we can talk offline about that, because we're generating, we're doing, as I said earlier, beginning, starting to do lots of these analyses on, looking at young and old and predisposition to cancer, etc. So
yeah, that'd be super cool. I'd love to talk.
All right, maybe we should call it. We're over time, but this was wonderful. Thank you so much for joining us, Jean, and thanks Peter and Jeff for helping to organize this seminar series. This talk was recorded. That's correct, Peter?
Yeah, it's recorded. Yeah, so it'll be posted on the website.
Okay, great.
Yeah.
And, do you know who the next speaker is, Peter? We
don't actually have anyone lined up for next month.
Interesting. Okay.
So if anyone has suggestions, please email myself and Pallav.
Yep, happy to talk more about that if anyone has suggestions. Thanks everyone for tuning in, and see you in the future. And Jean will connect offline.
Thanks.
And I'll put you in touch with Peter as well.
Thanks, Jean. That's really wonderful. Thank you.
No, thanks so much. Take care, everyone.
Bye.